Hey, my name is Dr. Rizky, medical student at Azhar University. And this video is about islets of Langerhans. The pancreatic islets are the regions of the pancreas that contain its endocrine cells. Discovered in 1869 by German pathological anatomist Paul Langerhans. The pancreatic islets constitute 1 to 2% of the pancreas volume and receive 10–15% of its blood flow. The pancreatic islets are arranged in density routes throughout the human pancreas and are important in the metabolism of glucose. Now let us talk about the structure of these islets. There are about 3 million islets, distributed in the form of density routes throughout the pancreas of healthy adults, each of which measures an average of about 0.1 millimeters, 109 microcm in diameter. Each is separated from the surrounding pancreatic tissue by a thin fibrous connective tissue capsule, which is continuous with the fibrous connective tissue that is interwoven throughout the rest of the pancreas. The combined mass of the islets is 2 grams. Islets of Langerhans can also form superstructures called islet clusters surrounding large blood vessels. The roundness of islets along the pancreas has also been quantified as an index of sphericity. Islets closest to the spherical form are mainly found in the tail of the pancreas, whereas the least spherical islets are found in the neck of the pancreas. Microanatomy. Hormones produced in the pancreatic islets are secreted directly into the blood flow by at least 5 types of cells. In rat islets, endocrine cell subsets are distributed as follows: alpha cells producing glucagon, 20% of total islet cells; beta cells producing insulin and amylin, almost equal to 70%; delta cells producing somatostatin, less than 10%, Page's cells; gamma cells or F cells, producing pancreatic polypeptide, less than 5%; epsilon cells producing ghrelin, less than 1%. It has been recognized that the cytoarchitecture of pancreatic islets differs between species. In particular, while rodent islets are characterized by a predominant proportion of insulin-producing beta cells in the core of the cluster and by scarce alpha, delta and Page's cells in the periphery, human islets display alpha and beta cells in close relationship with each other throughout the cluster. What is the function of these islets? These islets can influence each other through paracrine and autocrine communication. And beta cells are coupled electrically to 6 to 7 other beta cells, but not to other cell types. The paracrine feedback system of the pancreatic islets has the following structure: glucose/insulin activates beta cells and inhibits alpha cells. Glycogen/glucagon activates alpha cells, which activates beta cells and delta cells. Somatostatin inhibits alpha cells and beta cells. A large number of G-protein-coupled receptors, GPCRs, regulate the secretion of insulin, glucagon and somatostatin from pancreatic islets. And some of these GPCRs are the targets of drugs used to treat type 2 diabetes. Electrical activity of pancreatic islets has been studied using patch clamp techniques. It has turned out that the behavior of cells in intact islets differs significantly from the behavior of dispersed cells. Now let's move on to the clinical significance of islets. The beta cells of the pancreatic islets secrete insulin, and so play a significant role in diabetes. It is thought that they are destroyed by immune assaults. However, there are also indications that beta cells have not been destroyed, but have only become non-functional. Finally, transplantation. Because the beta cells in the pancreatic islets are selectively destroyed by an autoimmune process in type 1 diabetes, clinicians and researchers are actively pursuing islet transplantation as a means of restoring physiological beta cell function, which would offer an alternative to a complete pancreas transplant or artificial pancreas. Islet transplantation emerged as a viable option for the treatment of insulin-requiring diabetes in the early 1970s. With steady progress over the last 3 decades, recent clinical trials have shown that insulin independence and improved metabolic control can be reproducibly obtained after transplantation of cadaveric donor islets into patients with unstable type 1 diabetes. Islet transplantation for type 1 diabetes currently requires potent immunosuppression to prevent host rejection of donor islets, an alternative source of beta cells. Such insulin-producing cells derived from adult stem cells or progenitor cells would contribute to overcoming the shortage of donor organs for transplantation. The field of regenerative medicine is rapidly evolving and offers great hope for the nearest future. However, type 1 diabetes is the result of the autoimmune destruction of beta cells in the pancreas. Therefore, an effective cure will require a sequential, integrated approach that combines adequate and safe immune interventions with beta cell regenerative approaches. It has also been demonstrated that alpha cells can spontaneously switch fit and transdifferentiate into beta cells in both healthy and diabetic human and mouse pancreatic islets, a possible future source for beta cell regeneration. Thank you. Bye.